Ever wonder how your environment can change your genes without altering your DNA? Welcome to the world of epigenetics. Step 1. Environmental factors like diet and stress can influence gene expression. Step 2. During different developmental stages, your genes express differently. Epigenetic mechanisms like DNA methylation and histone modification are key players. These changes can affect disease susceptibility and development. Want to dive deeper into how your lifestyle impacts your genes? Explore the fascinating field of epigenetics today. Unlock the secrets of gene expression and environmental influence, or stay in the dark. Your choice. Genomics is a fascinating field that dives deep into the complete set of genetic information in organisms, known as genomes. It's all about understanding how genes work and interact, and why genetic variation exists between individuals and species. Imagine personalized medicine, where treatments are tailored just for you based on your unique genetic profile. Or think about genetic diagnosis, which helps identify disorders before they become serious. And then there's synthetic biology, where we design new biological systems. Within genomics, we explore structural genomics, functional genomics, and comparative genomics, each revealing more about the intricate dance of life at the molecular level. It's a thrilling journey into the blueprint of life itself.
Genetics. It's the blueprint of life, shaping who we are. Scene 1. Heredity. The magic of passing traits from parents to offspring. Scene 2. Variation. Why no two of us are exactly alike, even twins. Scene 3. Genes. Tiny segments of DNA with big roles in our traits. Scene 4. Genetics helps us understand diseases and characteristics. Scene 5. Curious about DNA or inheritance? Dive into genetic science.
Ever wondered about the branches of genetics? Let's dive in. First up, molecular genetics. It deciphers genes at the molecular level. Then population genetics. It studies genetic variation in populations. Mendelian genetics focuses on inheritance principles by Gregor Mendel. Epigenetics explores gene expression changes without altering DNA. And genomics, it examines the structure and evolution of genomes. Each branch offers unique insights into the world of genetics. Curious to learn more? Let's explore the fascinating world of genetics.
Molecular genetics is like the blueprint of life, unraveling the mysteries of our DNA. From understanding DNA structure to gene expression, it's a deep dive into what makes us, us. Imagine editing genes with precision tools like CRISPR. It's like rewriting the code of life. This field is revolutionizing genetic engineering, gene therapy, and even personalized medicine. Curious about how these advances are shaping the future? Let's explore the latest in molecular genetics. Mendelian genetics reveals the fascinating rules of inheritance that shape who we are. The law of segregation tells us that each pair of alleles splits during gamete formation, ensuring genetic diversity. Then there's the law of independent assortment, which explains how different genes are shuffled independently, like a deck of cards. And let's not forget the law of dominance, where one allele can overshadow another, determining traits like eye color. Understanding these principles not only helps us predict traits, but also sheds light on genetic disorders, making genetics a key to unlocking the mysteries of life.
The law of segregation is a fascinating principle in genetics. It tells us that each pair of alleles separates during gamete formation, meaning each parent contributes one allele to their offspring. Imagine this. During meiosis, alleles for a trait split apart, resulting in gametes, sperm or egg cells, that carry just one allele. This process is crucial for understanding how traits are passed down through generations. It allows us to predict the likelihood of certain traits appearing in offspring, shaping everything from eye color to genetic disorders. The beauty of Mendelian genetics lies in its simplicity and its power to explain the complexity of inheritance. The law of independent assortment reveals a fascinating truth about genetics. Alleles for different genes are sorted independently during gamete formation. This means that traits like eye color and height are inherited separately, allowing for a unique combination of characteristics in offspring. Imagine a child with blue eyes from one parent and tall stature from another. Thanks to this law, it's a key principle that drives genetic variation and diversity, helping us predict how traits are passed down through generations. Understanding this can unlock the mysteries of inheritance and the rich tapestry of life.
Ever wondered why some traits just seem to dominate? It's all about the law of dominance in genetics. Step 1. A dominant allele shows up if you have one or two copies. Step 2. A recessive allele only shows if you have two copies. This helps us predict how traits are expressed. Want to dive deeper into Mendelian genetics? Let's explore how dominant and recessive alleles interact. DNA is an incredible double helix made up of two complementary strands twisted together. These strands are built from nucleotides, which are the building blocks of life, consisting of sugar, phosphate, and nitrogenous bases: adenine, thymine, guanine, and cytosine. The beauty lies in base pairing. Adenine pairs with thymine, and guanine pairs with cytosine, creating a perfect match. During replication, the double helix unwinds, and helicase separates the strands, forming a replication fork. DNA polymerase then steps in, adding nucleotides to the template strands. The leading strand is synthesized continuously, while the lagging strand is made in fragments known as Okazaki fragments. Understanding this process is essential for grasping genetic inheritance and the fascinating world of genetic engineering, where we manipulate DNA for groundbreaking applications.
Gene expression is a fascinating process that transforms DNA into functional proteins. It all starts with transcription, where DNA is converted into RNA. Then, during translation, that RNA is used to build proteins, the workhorses of our cells. But it's not just a straightforward process. Regulation is key. Transcriptional regulation controls when and how much RNA is made, while post-transcriptional regulation manages RNA processing and stability. Epigenetic factors like DNA methylation and histone modifications add another layer of control. This precise regulation is crucial for cell differentiation, development, and even in diseases like cancer. Plus, it opens doors for innovative applications like gene therapy and synthetic biology, where we can design new gene regulatory systems to tackle health challenges. The world of gene expression is truly a game changer. Mutations are fascinating changes in our DNA, like point mutations that tweak single nucleotides, or chromosomal mutations that shuffle entire chromosome structures. They can arise from errors during DNA replication or from environmental factors like radiation and chemicals. Thankfully, our cells have robust DNA repair mechanisms. Base excision repair fixes damaged bases, while nucleotide excision repair tackles larger lesions. Mismatch repair corrects replication errors, and double-strand break repair mends severe DNA breaks. These processes are crucial for maintaining genome stability and preventing diseases like cancer. When DNA repair goes awry, it can lead to genetic disorders and increased cancer risk. Understanding these mechanisms is key to unlocking the mysteries of genetics.
Point mutations are fascinating changes in our DNA, where just a single nucleotide can make a big difference. Think of substitutions where one base swaps for another, like A turning into G. Then there are insertions adding an extra base and deletions which remove one. The effects? They can be silent, causing no change in the amino acid sequence, or they can lead to missense mutations, altering the protein's structure. Sometimes they even create a nonsense mutation, leading to a premature stop codon and a truncated protein. These mutations can cause genetic disorders like sickle cell anemia or cystic fibrosis. influence our susceptibility to diseases, and even drive evolution by introducing genetic variation. It's incredible how such tiny changes can have such profound impacts.